Good morning again. Hello, I'm Jenna Rivlin, and my bioinformatics class this semester is doing a project with Hudson Alpha called the CODE project, where we look at different gene variants and then make some hypotheses about their mutations and the effect on the proteins of the cell. This is Elin Zaman, and she's going to be giving you a presentation on one of these particular variants.
Hi, my name is Elin Zaman, and I've been conducting research on an exploration into the effect of the T147A clinical VUS in CTLA4 with Dr. Rivlin, as well as the CODE program with Hudson Alpha Huntsville. The CTLA4 is a gene which is located at chromosome 2 on the Q arm on the 33.2 position. It is also a known cytotoxic T lymphocyte-associated protein 4. Cytotoxic being toxic to cells such as immune cells and types of beta. T lymphocyte is a type of white blood cell which is part of the immune system. The function is damaging infected host cells as well as activating other immune cells and regulating the immune response. The CTLA4 is among the immunoglobulin family, which produces a T cell inhibitory protein. The gene is also a protein receptor which functions as an immune checkpoint and downregulates the immune responses. This is extremely vital to the immune response in the body. The pathological phenotype of the CTLA4 gene has been implicated to influence susceptibility to autoimmune diseases such as insulin-dependent diabetes, type 1 diabetes, which is where the immune system attacks and destroys insulin-producing cells in the pancreas, as well as Graves' disease, which is an autoimmune disorder with the overactivity of the thyroid gland and hyperthyroidism, as well as Hashimoto thyroiditis, which is similar to Graves' disease, where the immune system attacks the thyroid gland. It also causes hyperthyroidism. Celiac disease, which is an immune reaction to eating gluten. It triggers an immune response in the small intestine, and lupus, which occurs when the body's immune system attacks its own tissues and organs. This is a graph of the variant impact of the CTLA4 gene. The blue on the chart is a benign variant, while the reds are the pathogenic variants. My variant is in the 147 amino acid position, and it has a CAD score of 3.60. The CTLA4 variant, my CTLA4 variant is a missense point mutation, which is where a single nucleotide changes results in a codon, which codes for a different amino acid. My variant had a nucleotide change of ACC to GCC, where the adenine had been swapped to a guanine. This resulted in an amino acid switch from threonine to alanine at position 147. As you can see here, this is the structure of threonine. The amine group is the NH2, while the carboxyl group is the double bond to the O and the OH. While alanine is also has an amine group and the carboxyl group of the double bond to the OH, the double bond to the O and an OH. The difference between them is that threonine has a methyl group as well as an OH bond, while alanine only has a methyl group and a hydrogen. Threonine is a polar hydrophilic molecule, while alanine is a hydrophobic molecule. The variation location domain. The CTLA4 contains 3 domains. The V extracellular domain, which has the positions of 36 to 161 position, the transmembrane domain, which is the 162 to 182 position, and the cytoplasmic tail, which is 183 to the 223 position. My mutation, the T147A mutation, falls into the 36 to 161 position, which makes it extracellular, which means it's in the outer membrane of the cell organelles and cells. This is an imagery that was pictured with USARA, and the top image is my wild type, which has the T147 mutation in it, while the bottom picture is my variant, which has the switch from the T to the A. T147. This is a close-up of the T147 change to the A147 change, where you can see the side groups in the mutation, which is colored purple, where theriodine has the extra OH group, while alanine does not. These are my polyphen 2 results, which had shown that in the Hum div score, it was a 1, while the Hum bar score had been a 0.9. Polyphen 2 is a bioinformatics tool which predicts the possible impact of an amino acid substitution on the structure and function of a human gene. The fact that my results have been probably damaging shows that it most likely has a negative effect on the expressed gene. The Hum div score is on the Mendelian disease variants, while the Hum bar goes off of the human variants. This is my multiple sequence alignment that had been occurred. Multiple sequence alignment is of 3 or more biological sequences of a similar length. As you can see in the black box that shows T, that is my amino acid change, and the rest of these positions are similar organisms which have this variance inside their CTLA4 gene. These are my T147 scores. So my raw CAD score had been a 3.16, which the raw CAD score comes straight from the model. However, the values of the raw CAD score only define the rank of the variants of the gene. My CAD score had been 23.9, which is determined to be deleterious. This is on a scale, and it's more accurate of the determination of having deleterious effects. My CIF score had been shown to be damaging, also known as intolerance. So it's most likely between the 0.0 to 0.5 range. The CIF predicts whether the amino acid substitution affects the protein function. The fact that it is damaging means it most likely affects the protein function. My GURP-S score, which is a genomic evolutionary rate profiling score, it's a method to determine or estimate the evolutionary constraint and conservations. A higher score typically indicates more evolutionary constraints. The scale of this is a negative 12.3 to 6.17. My score had ended up being 5.34, which means that it most likely has evolutionary constraints. These were my citations. In conclusion, through exploring the variant of the T147A in the CTLA4 gene, it has been observed to be most likely pathogenic and cause negative connotations to the gene structure and function. Thank you. Any questions? Yes.
I know you have a lot of collaborative arrangements with other, but how much of this research can actually be done on campus, like just in-house?
On campus, well, all of this research is done on the computer, actually. So it's through multiple programmings. So if anyone was interested in it, they could talk to Dr. Ridley and ask about it. We use multiple programming such as Yasara, ClinVar, Polyphen 2, and all of these can be easily accessed through a simple Google search. And there's multiple variants of certain genes that have never been explored. So we're simply taking certain mutations to determine the pathogenic effects of this CTLA4 gene.
So there's basically a public database.
There's a big public database
Okay.
that everyone has access to and can look into.
Thank you.
Yes?
I'm assuming that this gene in question isn't one that typically is tested as part of, say, an ancestry DNA or something like that test, because they tend to be slices of the genome. So
Yeah.
what resources are available for whole genome sequencing nowadays?
I'm sorry, I don't really know too much information about that. I haven't looked into that particularly, So I'd have to look into it a little more, but I could get back to you and look into it.
All right, thank you.
It's, can I kind of scoff about that? It's really expensive, so, if you just do a Google of DNA tests, you can find tons that are pretty cheap, including 23andMe and all the ancestry ones. But if you want a whole genome sequencing, there's still only a few companies that do that just for folks who buy it and not with a clinical indication. And oftentimes it's hard to get insurance to cover it if you even do a clinical. So it's a good question, especially for genetic counselors. I think they'd be able to tell you like an exact price point. But the last time I heard how much it cost, it was in the thousands of dollars.
Okay, any other questions for our speaker?
Yes.
At the risk of sounding like I wasn't paying attention, because I was, it's mostly because I'm dumb. What does all this mean? Like, as far as like what it actually does?
So my mutation of the T147A gene, what we're determining here is whether it is pathogenic, so it can be likely to be seen in the, here, let me, really quick. It can be seen to cause susceptibility to the autoimmune diseases. So if you have type 1 diabetes or Graves' disease, it's possible that you might have a T147A mutation
Oh,
in the particular gene, which is CTLA4.
okay. Yeah, I must have my bypass for that, yeah.
It's alright.
I'm sorry. Thank you.
All right, well, let's thank our speaker again.